Hey there, students! Today, we're diving into the world of cell division, and guess what? We're doing it with claymation. Let's get started. So, cell division, what's it all about? Well, it's the process by which a single cell splits into two or more cells. It's super important for growth, repair, and making new life. There are two main types of cell division, mitosis and meiosis. First up, let's talk about mitosis. Mitosis is all about growth and repair. Think of it like photocopying a page, making identical copies. Mitosis kicks off with interphase. It's not technically part of mitosis, but it sets the stage. Here, the cell grows and duplicates its DNA, creating two sister chromatids. Next is prophase. The chromosomes condense and become visible. The nuclear membrane starts to break down and spindle fibers form, attaching to the chromosomes. In metaphase, the chromosomes line up at the cell's equator. Spindle fibers ensure each sister chromatid is connected to opposite poles. Then, in anaphase, the sister chromatids are pulled apart to opposite ends of the cell. Each pole gets an identical set. Telophase follows. Chromosomes begin to decondense, the nuclear membrane reforms, and the spindle fibers disappear. Finally, cytokinesis. This is where the cytoplasm divides, pinching the cell into two. And voila, we have two identical daughter cells. Now let's switch gears to meiosis. Meiosis is a bit different. It's for making gametes like sperm and egg cells, and it ensures genetic diversity. Unlike mitosis, meiosis involves two rounds of division. First, meiosis 1. Prophase Y is where chromosomes condense and pair up, and crossing over happens. They swap bits of DNA, creating genetic variety. Then in metaphase 1, homologous chromosomes align at the middle of the cell. Anaphase, I sees these pairs pulled apart to opposite poles. In telophase Y and cytokinesis, the cell divides into two haploid cells, each with one chromosome from each homologous pair. Next, meiosis 2, it's similar to mitosis. Prophase 2 has chromosomes condensing again. In metaphase 2, chromosomes line up at the equator of each cell. During anaphase 2, sister chromatids are finally pulled apart to opposite ends. And in telophase 2 and cytokinesis, we get four unique haploid cells. So what's the big difference between mitosis and meiosis? Mitosis is like making identical photocopies, perfect for growth and repair. Meiosis, on the other hand, is like shuffling a deck of cards and splitting it in half. Each resulting deck is unique, which is perfect for making gametes. To wrap it up, mitosis means two identical diploid cells for growth and repair. Meiosis means four unique haploid cells for sexual reproduction. Alright, that's it for today. Thanks for watching, and remember, keep learning and keep growing. See you next time.